the problem we're looking at. I know for a fact that a lot of people are working on detection for this method. And I think clinically, diagnostically, a lot of people are using. They have to culture the bacteria, but you'll get a lot of E. coli from the patient. So, and then if you want to differentiate salmonella or other bacteria, you have to do different types of cultures on different plates and distinguish them. That will be time consuming. So the best method is to use our molecular tools to do it, and we can do that using PCR, and then we are looking at specific genes. So I chose RFBE gene because that is commonly associated one of the target when you want to detect the most harmful version of the E. coli, which is E. coli 0157H7, if I'm not mistaken. And when there was some papers talking about that, and then everybody's using different types of primers, and they verify their primer whether the primers were working or not, right? So so that brings back to our problem, which is now we have to design a primer using RFBE gene. The problem is that after that, I got a bit excited, and then I decided to add a few more organisms, which doesn't play very well when you align them inside. So most of you, let's look at what I did. All right. So I just I forgot the gene. RFBE, right? All right. RFBE in nucleotide, right? So you'll get a lot of you will have no problems here because our main focus last week was to learn how to use the database. And then where did I get the idea to add extra organisms? From here, right? On the right hand side, you can see there's E. coli, there's Mycobacterium, there's Salmonella, and then I add Vibrio because I know Vibrio is a common Enterobacteria pathogen, so I added Vibrio inside. So if you click here, top organisms, so these are, well, they give you a lot of, let's go one step back so that we can look at the difference. Oh, let's go back. Right, all right. So you get E. coli here. So the first one, let's collect all the sequences. If you just click E. coli, oh, let me just. So if you add E. coli, you click E. coli, then you'll get the gene for E. coli. It depends on how you arrange. If you sort by length, you get different ones. Let's go by default order, and I think you can find the gene there. But 242 base pair is not the actual gene, so you have to find which one is the correct ones. So I think the lesson from here is for you to identify which sequence to choose, which is very important. And you see here, partial CDS means it's not the full gene. It's just part of the CDS means coding sequence. So that's not the full gene. So I think you have to look at for the complete gene, and over here you can find, for example, Contic 45. So this is from genome assembly. If you click that one, this is about 15 kilobase. That's very big. And I think from the Contic here, you can find RFEB, right? Yeah, RFBE, right? All right, RFEB, right? Okay. No, I got confused. RFBE, right? So you can Control F, and then you can find the sequence over here. So one of the option that I gave you, but nobody only wants to use that option, was to look at intra intergene and intragene, right? So the gene, let's just draw the gene as a line, right? So you can have the gene start from ATG, and then let's end it with TGA or TAG or TAA. So this is the common universal stop codon here. 3 options over here, but normally you'll start with ATG, and most of you will try to find the consensus, right? When you get you align using your cluster omega, you align Vibrio, Salmonella, and then there are slightly different lengths, right? So and then you'll get some consensus, and because microbacteria was not giving a good alignment, some of you would design 4 sets of primers, but I was hoping that someone would use the region here, which is before and after gene. To be able to do that, you have to choose the right sequence over here, because you have to not just look specifically for the gene, but you have to look at a wider like this contig over here, so that you'll get the upstream and the downstream sequence, hoping that maybe they align better than what's inside the gene, right? But apparently, even if you do that, it doesn't align very well. But so there is no one correct answer, all right? There's no one correct answer. And if you have to do 4 sets of PCR to determine different types of organisms, then maybe that's what you have to do. And for the third part of the answer, a lot of you answered using qPCR, multiplex PCR, which is also correct, but there are difficulties in doing that. Because for genetic engineering here, this class, we're not talking about the experimental difficulties or limitations. Like designing a multiplex PCR assay is very complex. And regardless of what you design here, you have to validate it by experimentation. That's why there's a lot of papers describing their efforts on using different types of method to determine to detect different types of pathogens. And also, if you think out of the box, you can use different methods, not just this particular gene. You can use this gene to detect whether it is related to the O antigen, and you can use another set of primers based on maybe 16S ribosomal RNA, which is commonly used to detect different types of bacteria. You can use also repetitive sequences or other regions, like for fingerprinting of different types of pathogens. So I think, especially for UPM students, I think you did quite well, and a lot of you really put effort in answering the question. Although I think the most difficult part is aligning it with cluster W. And also one more thing, not many use the was it the nucleic acid notation, right? The nucleic acid. Let me just Google that. You can find it in Wikipedia to notation, right? Okay, so you get the chart over here. So if you have to design a primer that uses at a specific location like C and G, either C or G, you can replace that with an S, right? So if you have A and T, you can use W, right? Use W. And then if this is for the basic ones, the ATCG is for a single option. We don't use U because U is for RNA. But if you have multiples, right? Multiples, you can use this one. So for universal primers, sometimes we use M because that location can be an A or a C, right? So you can replace that with an M. So when you actually order the primers from the supplier or the manufacturer that produces the primers, you just specify M, and they will add A and C, equal amount of A and C at that particular location, so that that primer would be able to enable or bind to the location with A and C. So these are very useful. WSMK, all right. And then if you have a multiple of CGT, right, over here, like more than three, so you can use these letters. I seldom see people use these letters, but maybe sometimes you need to use that because you cannot align them very well. Another approach, if you cannot align them very well, I think Vibrio and E. coli aligns very well. You use the same primer for Vibrio and E. coli, and then after that, you can design a separate primer for Mycobacteria and Salmonella instead. Another approach, if you want to have the whole PCR reaction in one tube, you can design different primers that will give different size product, right? Different size product. So you can have 400 base pair for 300 base pair for E. coli and Vibrio. You can have 200 base pair for Salmonella. You have 100 base pair for Mycobacterium. So when you can run it in the same tube if you do multiplexing, and then when you run the gel, you can see in one lane you can determine whether or not the gene is detected, is available, and the second information you get from that is what type of organism that you have. So there's a lot of room to play, and that's why there's no single solution. And another reason why there's no single solution, depending on which result you choose, you might get different types of primers. So I cannot give you one primer, and then even for you to look at which region I chose is quite difficult. So in the end, it's yeah, there's a lot of variables in the types of answers you can submit. All right. So today we'll move to the next step, which is let's move from just looking at individual genes and then looking at we did look at individual genes, design primer for individual genes. Last week was looking at multiple genes from different organisms, trying to find consensus within them, and then have our own primer, design a primer so that we can detect and distinguish them at the same time. And now we'll be having some procedures using plasmids and the DNA cloning over here. So I think we had enough with E. coli and Vibrio and Salmonella. Let's move back to SARS CoV-2, right? COVID-19 virus. So the process of detection of SARS-CoV-2, first of all, because it's an RNA virus, the RNA must be converted to DNA, right? So that we can work with PCR. So the process involves reverse transcriptase, right? Using reverse transcriptase. We will reverse transcribe the RNA, it becomes cDNA, and then using specific time, amplify them, and then we get a lot of DNA, because we usually don't get a lot of virus. So we have small amount of RNA, and then converting the RNA into cDNA will not necessarily produce DNA, but only after the typical PCR step, you'll produce a lot of DNA, right? So when we have a lot of DNA, we have a lot more samples to work with, so it's easier for us to proceed whatever we need to do. For the typical case, if we have a new variant, we want to sequence the gene, and then we can determine whether now we have another mutation in COVID-19, then we need another booster shot of a vaccine or so on, something like that. The previously I explained about what I know, at least the typical uh, uh, anatomy of the SARS-CoV-2. We have the spike protein. We have here the nucleocapsid, the membrane, the envelope, and then the single-stranded RNA inside. So to detect this using PCR, of course, our target will be the genetic material. So we want the RNA, and then we can convert them into DNA for our next work. So if you remember back in 2020, when everybody's panicked and everyone wants to get hold of a method to detect, quickly diagnose whether you have COVID or not, we ran out of RNA extraction kits, right? Because RNA extraction kits used here to extract the RNA. And I think most popular brands, even non-popular brands, everybody's buying them, and then not many people are producing it enough for this purpose. And I think RNA extraction kits are normally used for research purposes. All right, so this is not much for clinical or diagnostic purposes. So I don't think the supply is a lot, and when there is a rush of orders coming in for RNA extraction kits, even for people not doing work for COVID or people doing research for other purposes, we cannot just we cannot buy enough kits for doing that. So extracting RNA is somewhat similar to extracting DNA. And if you're familiar with kits, there you have a box. If for me, I'm working with bacteria, so there'll be some buffers you need to add to break the cell, and then after that you have to separate the lipid, separate the protein from the nucleic acid, and then within the nucleic acid, if you add DNase is an enzyme that will break down DNA, and the remaining product will be RNA. And then after that, you will have some buffer washes to extract your RNA from the solution. So previous activity we did, this was our first activity. We did design the SARS-CoV-2 primers, and then what we did was, this is slightly different. We get the information from the gene bank, and then we designed the forward and reverse primer, and then we did test it with PCR. So today we will add the element of plasmid so that we can clone our sequence in plasmid. Hold on, I'm not getting my slides here. Okay. Let me refresh. If maybe I should download. I got logged out from my screen. Hold on. Let me. I wonder what happened here. Let me just download it. Wow. I think my computer doesn't like the class today. All right. Let's share the screen again. Okay. All right. So we did tested out the PCR simulation. So today, the difference between previous classes, our aim was to look at how to get genes, how to identify genes, how to identify ORFs, and today we will, only for the first time in this class, genetic engineering, we will try to manipulate these DNA molecules. So first of all, we need the genes, right? So we know, I mean, for the past 3 classes, we know how to obtain genes from the gene bank. the information. But in the lab, all right, in real life, what we need is first of all, you can get the gene from an organism, all right? So if you're working with bacteria, you can get the culture uh, from different sources. You can buy the strain, you can isolate from the environment, you can grow the bacteria, and then you can extract the DNA, like I mentioned just now. Other than that, of course, if you're collaborating with other people, you can get the DNA from them, so you don't have to handle the bacteria. So that is very important because sometimes you're working with pathogens, right? And your environment, your lab is not necessarily equipped to handle harmful organisms. So dealing with DNA is a lot safer. So you can have the medical faculty from a different university who has the equipment to handle, let's say, Ebola, for example. So they can handle Ebola viruses. And then after that, you get the DNA. DNA cannot infect people, right? Because it's just the nucleic acid. So you have the gene. You can then after that you can easily manipulate and do research on it. So that is one of the advantages of dealing with DNA molecules, so that you can do research without the risk of handling dangerous organisms. And other than that, if you're working with human DNA, for example, so sometimes you cannot get the organism because the organism is human, right? And that's when working with genes makes a lot more sense and making impossible things possible. So what do we do when we manipulate genes, all right, the DNA? So first of all, we can copy, right? We can copy. I mentioned just now, where if you have a lot of sample, it's easy for you to work with because at low concentration, DNA tends to just disappear, right, in your test tube if you have a very low concentration, because every time you manipulate the liquid, for example, you transfer liquid from one container to another container, you will lose a bit of material. And if you do, for example, you need few microliters of DNA to run electrophoresis, and you'll be wasting few microliters from there. So you are losing things. But because we have PCR, we can always make more, right? And to be able to make more, you need to have the right primers. Hence why we learn how to make primers, because we can get small amount of DNA, we can multiply them and produce more DNA. And that's very practical when we have to do a lot of things. And when you have a lot of DNA, when you're collaborating with other places, and when they want a bit of a sample, you don't feel very reluctant to give them, because you can give them as much as they want. And then if you're running out of that piece of DNA, you can always reproduce them using PCR, right? So copying or amplifying DNA is very important. And then after that, when we want to clone, right? So I'll talk about cloning. We will need to cut things, right? And then we need to rearrange them, all right? And cutting involves using this nuclease enzyme, right? Digesting, the process called digesting. And basically, we just refer to nuclease as restriction enzymes. So we will cut it and then we will attach them again. That's the rearranging process. And joining two molecules of DNA together is the process of ligation using the enzyme ligase. So this PCR, you use PCR enzymes, Taq polymerase, nuclease, restriction enzymes, and ligation. So these are the three basic enzymes, three basic tools that we use in the lab to manipulate them. So you can get these enzymes very easily. And for example, I always use in Japan here, we use Nippon Gene. So I think normally you heard of New England Biolabs, NEB, for example. And then you have companies like Takara here in Japan, also is very famous brand. I use Nippon Gene because I find it's tend to be a little bit cheaper than other big brands. But Nippon Gin is also quite a good brand. And when you go to the website like this, you can see tools for research, reagents for diagnosis, detection, human animal IVD. But I often go here, and in Japan, normally about twice a year, right, during summer and now winter, where they have winter sale, right? So you have winter campaigns for these kind of products, and usually they give you about 20% to 30% off from the normal price. And here, just to look at the price, the website is let's go in English. But let's go in English and see. Somehow, if you go to the page in English, there's no more sale, right? So I think that's what happens in Malaysia. Usually, you'll get the normal price. So enzyme and protein. Let's look at PCR. So PCR attack polymerase. So here, even the price is not available in English. All right, let's just go back into Japanese page. Let's look at real-time PCR. Let's look at restriction enzymes instead. There is no real-time PCR, but there's no normal PCR. It should be. All right, let's just look at restriction enzymes. So it's about 5,400 years, about maybe 200 ringgit per tube of restriction enzymes. So there's a lot of different types of restriction enzymes. Where can we find a whole list of restriction? So you can click here, and this is the campaign they're doing. So over here, there's different types of restriction enzymes with different prices. So you can buy this, I think, fairly easily, and you don't have to be in a chemical laboratory. For example, if you need to buy chemicals like solvents and so on, they won't send things to your house because chemicals are dangerous. But enzymes are different things, right? So, but unless they find that they don't want to send it to your house for some reason, then you can buy it. But normally enzymes are not categorized as harmful materials like chemicals. They are not acids or they are not corrosive materials. So anybody can buy them. Hence, there's a group of people, I think, around the world are doing this as a hobby. They do DNA cloning in their garage. For example, they convert their garage into a lab, and then they play with it. Well, actually, the sale is a lot better, 40% to 50% off, right? So then because people are doing this as a hobby, then there's the term synthetic biology, hobbies, all right? So people are playing with DNA sequences and PCR as a hobby, right? So I find it not very weird because it feels like, doing puzzle solving or something like that. But I think if you can get the material quite cheaply, you can do it. You can do it at home, I think, without any much problem. Of course, you need PCR machine. You need electrophoresis device apparatus to do that. But those things are not expensive as well. So let's go back to our lecture. So you can get the PCR enzyme, nuclease nuclease enzymes, and then the ligase enzymes readily from suppliers, whichever brands you like. And this, if you know how to use them, it's quite easy. And in this class, we're learning about the theories of things, but practically it's also not that difficult. Right? So DNA cloning is another word for it is the recombinant DNA technology. So the word clone always means to make copies, right? To make copies. But in molecular biology, here we use DNA cloning to produce something else, not just copies, but we still copy part of the original genetic material, which is the insert, right? So normally we'll use plasmid, and then that's where we focus today. We have plasmid DNA, and then we will cut the plasmid DNA using our nuclease and insert the piece of DNA that we have. So that piece of DNA typically we will produce using PCR, and to produce using PCR we need to use primers. And then after that we can insert this plasmid into our organism of choice. For me, I use bacteria, I use E. coli, because E. coli is the most, I think, easiest to do compared to other organisms, because it's a bacteria, prokaryotic, very simple, and they grow very fast. And then after that, we can extract the plasmid again from them. So depending on the type of plasmid, there are high copy plasmids, which means that once you insert the DNA into the E. coli, it can self-replicate, right? It will self-replicate to many copies. Some high copy plasmids can replicate itself 700, 800 times inside each cell, right? So when you grow bacteria on a plate like this, so a single spot here is one colony. So one colony originates from one cell, but it replicates itself into I think few thousand cells maybe. And then each single cell will carry 700, 800 copies of the plasmid, which means that if you pick up one colony, you'll have a lot of plasmids, right? A lot of plasmids. So that's one way of making copies of of DNA. And just now we were talking about using PCR to manipulate DNA. And when you want to copy DNA, we use PCR, right? And PCR, because we have a DNA polymerase enzyme inside, and it will repeat the process of copying, right? Copying the DNA. However, when you grow a cell, a bacterial cell, the bacterial cell will multiply. When the bacterial cell multiply, it will also multiply the amount of DNA it has, right? So this is more, I think, organic way or more natural way of DNA of copying DNA material. But to be able to do this, you have to clone it into the plasmid DNA in the first place, right? So we'll look at some steps to make this happen. So let's look at enzymes. Just now I explained. First of all, you have to get your DNA from some sources, just small amount of DNA, and then you can copy them using PCR. The enzymes we're using here is DNA polymerase, and previously, like mentioned, we use Taq polymerase for PCR. And then we have to cut them using nucleases, restriction enzymes. There's two types of nucleases, endonucleases and exonucleases. Exonucleases will cut DNA from the end, right? From the end. So there's a 5' here, 3', so it will cut it from the end. But because the tools that we normally use for DNA cloning will be endonucleases, so this will cut DNA in between, not the end, right? So the difference is at the end, you have phosphate groups, right? And then the 5', the lack of phosphate groups. But endonuclease will require those phosphate groups to be in the middle. That's how the DNA cutting process happens. And finally, the reverse process of nucleases will be ligases. So these enzymes will join together. So you need two molecules, or at least two ends of DNA, and this will complete the phosphodiester bonds, which is the phosphate groups joining two bases. This one, I don't think we have to repeat this, but for DNA polymerase to work, you need primers, and then DNA polymerase will begin the replication or copying DNA process by, first of all, synthesizing the new DNA strand from the 5' of from the 3' side of the primer according to the complementary strand, right? Which is the template, right? The template over here. So DNA polymerase will be able to replicate, make copies of DNA in between the regions of primers. So you need two primers, forward primer and reverse primer, and only the part in between will be copied a lot, many times, but not the rest, right? So in contrast, the method that when we use plasmid, all right, when we use plasmid, we can copy the whole plasmid regardless of primer because we don't need primer, right? So this is also an advantage of making copies using DNA, using the bacteria, because we are using the natural mechanism of bacteria to replicate the plasmid. So this is very important because like last week's assignment, you know that you cannot it's not easy to design primers, design primers when the sequence don't align, and then you need the information from the database to design primers. So what if when you are given an unknown piece of DNA, you don't know the sequence, you need to find a way to replicate it. So the best way is to put it inside the plasmid and then replicate it, and then after that manipulate it later. The DNA polymerase has different types of activity. I think we've talked about this before. 5' to 3', 3' to 5', 5' to 2' exonuclease activity. So these are different types of activity, and when you buy the polymer from sites like this, you can buy different types, right, depending on your application. So, So normally we'll have a 5' 3' DNA synthesis, all right, but those with 5', 3' exonuclease activity, this is slightly confusing because synthesis and exonuclease is cutting, synthesis is making DNA, but we have DNA polymers that have both activity, which is 5' to 3' DNA synthesis, but at the same time it has the ability to cut, all right, to remove base pairs in the 5' to 3' direction, which means that when the DNA polymerase comes in the middle, all right, it will have the ability to remove the base pairs downstream of sequence, right, from the location, for its location. And the B is the opposite, the 3', 5' exonuclease. This DNA polymerase has the ability to remove DNA from the opposite direction. So depending on how you clone this 5', 3' exonuclease activity and 3', 5' exonuclease activity are very important. So you need to use them to achieve some cloning methods. But the normal PCR enzyme that we often use for detection and so on is just the 5' to 3' DNA synthesis. Next is our reverse transcriptase. This you need, instead of using DNA as a template, now we need DNA as a template. So this is the type of enzyme that we use to convert RNA into DNA, right? RNA into DNA, and the DNA produced from a reverse transcriptase process known as complementary DNA or cDNAs. So if you go to GenBank and search for, I think most of eukaryotic organisms genes, they are often labeled as cDNA rather than just DNA, right? Because it is obtained from RNA and then it was reverse transcribed to become DNA, right? So the process of reverse transcriptase, this is example. You see there's a poly A tail, which means that it's from eukaryotic, it's not from bacteria. You can use a poly T primer, right? And this will produce a DNA based on the RNA template, right? So you can use an oligo DT primer, or you can use a random hexamer primer. So this is a primer, 6 hexamer, which means it's a 6 base primer. And because it's random, which means that how do you order a random hexamer primer? You just put the N, right? Because N represents ATCG, so it's totally random. And it will have the ability to bind at random location on the primer, right? So this is useful when you extract a lot of RNA and you want to convert all of the RNA to become DNA so that you can sequence them or clone them later. So we just use random hexamer. So I'm working with bacteria, so I don't have poly A tail like this because it's bacteria. And I have to use random hexamer all the time to detect specific genes that I want. And then if you know a specific sequence that you want to detect from your RNA, that's where you use your specific primer. So to date, we've learned a lot of we've learned how to design primers. So you can design primer to reverse transcribe your RNA to become a DNA, cDNA. Right? Next, this is the I think the focus for this week's lecture is exonucleases and endonucleases. So we're going to talk a lot about endonucleases, making internal cuts internal phosphodiester bonds, and we normally refer them as restriction endonucleases or RE, right, or restriction enzymes. So RE has many applications to generate fragments of DNA if you want to detect them, to prepare the end of sequences so that you can clone them. So those are the typical usage, right? Other than that, there are specific nucleases like nucleases S1 for single-stranded DNA, which is obtained from fungus. So this is for transcribe mapping, right? This is very specialized. And then the deoxyribose nuclease 1, which is specific for single and double-stranded DNA, also for nucleus footprinting, right? For fingerprinting to produce fragments. So the type of RE that we're using, there's a type 1 and type 2. Type 1, type 2, or type 3. There's 3 different types, but I think it's not necessary for you to know the difference of these 3 types. Basically, what it produces, there's 2 types of that it will produce. First of all, the blunt end type, and then the sticky type of they call it the blunt end and the sticky end, right? So different types of enzymes will cut differently depending on the recognition sequence. So for example, we have the enzyme ALU1 over here. LU1 will cut straight, right here. So this is the double-stranded DNA. Will cut directly when there is an AGCT. From the opposite side is AGCT as well. So this is the feature of restriction enzymes. But some enzymes, for ALU1, this is a 4-base recognizing enzyme, 4-base recognition enzyme, maybe, right? And it will recognize 4 bases, right? 4 bases. Some enzymes are much more specific. For example, EcoR1. So this EcoR1 is a 6-base recognizing enzyme. So it will recognize GAA, TTC as the location to cut, right? For this case, it's a sticky end. It will cut not in a straight manner, but will produce a protrusion, right? So this will become the sticky end. Why do we call it sticky? Because if you have a complementary end, right, another opposite complementary side of it, it will be able to stick to it quite well because ATCG and all the bases will form hydrogen bonds, right, readily when it's complementary. Other than that, like BGL1 here, you have ends, right? Ends over here. This is because at this location, it's a variable. You can bind anything, right? Anything there. And it will only recognize the GCC and the GGC at the far end, but not in the middle. So you can cut anywhere in the middle, right? So this, for this case, it will still produce overhang. So overhang and blunts are interesting because you can use them for different purposes. For example, when you cut something with, when you cut a piece of DNA with BAM H1, I call this BAM H1, it will produce a specific end that it will not bind to something that you cut with a R1, right? So it will not be complementary, so it will not join, right? So that is very good because you can use it to rearrange the things that you'll be cloning later so that you'll be in the right orientation. So here's the explanation. However, if you cut something with a blunt end, like example in C, right? This is sticky end, produce different enzymes. If blunt ends, you can always join with something else that is being cut with a blunt end as well, and that wouldn't be specific. The example number C in C is very interesting because now you can cut with BAMH1 and you can cut with SAO3A1, and that will produce the same overhang, right? And this will allow these two pieces of DNA fragments cut from different RE to join later, right? You can join them later with DNA ligase. And now let's look at DNA ligase. DNA ligase, the role in the cell is to fix the nicks, right? The nicks when you have when DNA is being cut. But in vitro, we can use this to join two molecules of DNA, right? So this is a blunt end joining to a blunt end. We can also use a sticky end joining to a complementary sticky end. So when you join the sticky ends together, this will form hydrogen bonds, and then the phosphodiester bond will be formed by the DNA ligase. So we can use this to add extra pieces of DNA because now we know how to cut blunt ends, and then we can join different molecules. We can buy linkers, right? Linkers. This small piece of DNA, we can join them to the end of our DNA molecules. Now you can have a modified piece of DNA with extra additional base pairs that is not originally there. So this is a solution of example. You can add these linkers that contain the sequence for your primers. You add them here, and now you can, after adding the additional sequences, now you can do PCR on an unknown piece of DNA, right? Because you don't know the sequence of the DNA that you get. Let's say you extract this DNA from the environment, and then you can add the linkers, and then after that you can do PCR from the linkers. Or you can add the linkers that contain restriction enzyme sites, and you can use that to clone afterwards, right? So this is very useful. And there are other enzymes that can modify the end of the molecules, just like adding the linkers just now. Those are alkaline phosphatase to remove phosphate groups. Once you remove phosphate groups, you cannot once you remove phosphate group, you need to use ligase to join the molecules, right? So this to prevent self ligation. So this is a bit technical. When the time comes. And then after that, there's polynucleotide kinase, perform a reverse reaction of alkaline phosphatase, adds 5' there, right? So this is the opposite of alkaline phosphatase. So it's not just removing the whole base, right, nucleotide, but you can also remove portion of it, which is the phosphate groups, which is important for joining the molecules. So for example, or the example is over here, so that's convenient. Why you do remove phosphate groups. For example, we have a piece of DNA which we want to add the insert, right? The insert, which is in red. But if we don't use the enzyme alkaline phosphatase, what will happen is that this piece of DNA, the plasmid, will join by itself because it's 5' here, 3' and 3', 5' over here. So it has a tendency of joining its end together and forming a complete loop, right? Complete loop. So when a complete loop is formed, there's no chance of a very low chance of DNA insert can enter here. So that is not a good thing for us. So to prevent the self-ligation of this long piece of DNA. We use alkaline phosphatase to remove this hydroxyl phosphate group so that it doesn't contain phosphate groups. And then only when a new insert is introduced, it has a phosphate group that can be completed by DNA ligase. So this is to prevent self-ligation. Excuse me. So today we're going to look at plasmids as vectors. So vectors as a vehicle to carry our piece of DNA. So we have, just now as explained, if you put plasmid inside the bacteria will replicate and the plasmid will replicate as well. So I mentioned just now, high copy plasmids will copy itself 700, 800 times in one cell. So you're going to have a lot of plasmid, right? And then after that, one of the merit of using plasmids is that you can put the DNA, all right, inside your piece of DNA that you want to study inside the plasmid. And the plasmid usually contain promoters, terminators at the end. And that will transcribe and then will translate, and then you can produce protein, right? That's how we produce recombinant proteins. And that bit we'll be talking about that next week. So for a plasmid, there's a plasmid. Usually they have this small letter P to indicate it's a plasmid. And then this particular plasmid is called PBR322. It's one of the, I think, oldest plasmid discovered in E. coli. So when you look at plasmid, you typically will be looking at a plasmid map over here. So PBR322 with the size of the plasmid, which is the base pair, 4361 base pair over here, and then you'll see restriction enzyme sites labeled, right, like EcoR1. That's at the location of 4359. That's the very end of the plasmid, right? And then there's HIND3, right? HIND3 at the location 29. So which means that location 1 is somewhere in between there. EcoR5, 167. So those are single cutting sites. Usually they will label that the single cutting sites on the outer side of the plasmid. Other than that, we can see other features. For example, the origin replication. So these are the sequence needed for self-replication of the plasmid. And then you have ampicillin resistant. So this is to select the E. coli when you grow them. And then you have tetracycline resistance genes over here. And then this is not a high copy plasmid. It just got 20 copies per cell. So it doesn't copy a lot. So when you do restriction enzyme cloning, right, we use restriction enzymes. This example here is we have EcoR1 here, and then we have other sites. I think we don't have NOT1, but this example below showing NOT1. You can choose your restriction enzymes of your choice, and then there's a criteria how you choose them. We'll see that afterwards. For this case, we'll do PCR, and then we'll add our restriction enzymes there using our primers, and then we'll cut and produce the sticky ends, right? We'll cut the plasmid site using the sticky ends as well, so that they'll have complementary ends. This one has EcoR1 and NOT1 being cut. And then by joining these two parts together and add ligase enzyme, we'll join them. We can restore the plasmid with the insert DNA inserted inside the plasmid. So the process itself is not just bioinformatics. We have to do, of course, lab work. We do the until ligation inside the test tube, and then we will need to insert this plasmid into the cell, right? E. coli. And inserting this process of insertion, you have two ways. You can apply heat using the heat shock method, or you can use electroporation. You apply an electric pulse. It will open the cell for few microseconds, I think, and then the plasmid in the solution will be able to enter the cell, and then after that, the cell will recover with the plasmid inside. And then you spread on the agar, and then you'll get a lot of colonies, and each colony should be carrying your plasmid, right? The reason is because it has the, for this case, for the previous case, ampicillin resistant gene, And the agar contain ampicillin, so only ampicillin resistant cells can grow on them. So that's how we do the selection. So for this class, let's look at PBR322 plasmid. Of course, we'll be checking them out in gene bank. All right. Okay, let's copy PBR322 over here and look for the sequence. So you get PBR322 cloning vector here, complete sequence. The size was 4361. So it is in agreement with our map just now, 4361, right? You can look at 4361. And let's click that and open. So this is cloning vector PBR32322. It was registered first in 1978, right? So that was a long time ago. And a lot of papers being written, and I think this plasmid has helped us understand a lot about how to do cloning in E. coli. If you scroll down, of course, you can see the sequence over here starts from number 1 to number 4361, if I was mistaken. The problem is the numbering, right? When you have a piece of DNA, you know the start codon, this is number 1, and this is number 400, 500, codon. But plasmid is circular, right? So we have to decide which is number 1 because it is continuous, it's circular. So sometimes the number could, depending on the data record that you get from the gene bank, you can get slightly different location for different genes. Let's click on the graphics. Right? This is a tool for us to visualize what are the features we have in the plasmid. So let's confirm we have ampicillin resistant genes. So ampicillin is an antibiotic. When this gene is expressed, it will produce a beta lactamase protein. This beta lactamase protein would be able to degrade or convert the antibiotic so that it becomes less harmful to the E. coli. The same goes with tetracycline over here. Tetracycline is also an antibiotic. So when this gene is expressed, it will disable the tetracycline effect, and because of that, the cell can grow even in tetracycline environment. So usually, resistance genes are written this way with a small r. Tetracycline tet and a small r, so to indicate that it's a resistant gene. So this as you can observe here, years ago we don't have this feature in gene banks. This is very easy to visualize. I wish that I had this feature when I was a student, but now it's a lot more easier. First of all, the part is the 2 genes that we can see here. All right. So if you put your mouse over here, that's BLA, which means beta lactamase. You can click on the blast record and so on. It will bring you to the comparison, the blast comparison for other genes. And then tetracycline is over here. And you can see the direction, right? Beta lactamase is from left, from left to right, right? So let's confirm that over here, our map, it doesn't show the direction, but we know it is facing opposite direction. So this is the start codon, this is stop codon, this is the start codon, and this is the stop codon over here. So direction is important, which also means that they are on a different strand of DNA. So this is on the what do you call it? I think coding sense, non-coding, sense or anti-sense, or the upper strand or the bottom strand, depends on how you refer to that. Over here, there's another protein, right? There's another ORF, open reading frame. There's a gene. It's a ROP protein. So that's for you can identify, although it's not really important for cloning, so it's not being labeled here. ROP protein. Other things that I find interesting, like maybe you can relate to, as already most of you studied molecular biology, you can see that to express a protein, all right, you need to have, let's go back over here, you need to have a promoter, right? Upstream of the gene, and then you need to have a terminator. So the promoter is where you get RNA polymerase binding to the promoter before transcribing your gene, right? Before copying your gene and produce RNA. So that is RNA polymerase, right? And after that, once you have RNA transcribed, there is a region up here that is required for the ribosome. It's called the ribosome binding site, right? And the ribosome will bind here and then will translate the RNA to become proteins, right? It will produce peptides, so it will produce polypeptides. So let's look at that. We know that we need to express this beta-lactamase gene upstream of that, right? You can look at here. This is the little binding features. If you put your not here, you put your here. You can find promoter here, right? So you can click on this gene and you draw a line. So just upstream of the beta-lactamase, there's a promoter P. So there's a promoter there for RNA polymerase bind, and then it will synthesize the RNA from the gene. And then after that, the ribosomal binding, or another name for ribosomal binding site is the SD sequence, Shine-Dalgarno sequence, right? So that's there. So the Shine-Dalgarno sequence must be slightly downstream from the promoter, because this will be part of the RNA as well. And then you can inspect also the direction of the beta-lactamase. So which means that this is upstream, Shine-Dalgarno is also upstream, and then the ATG of the operating frame will start here, and it will continue until, which is the size is about 861 nucleotides for the sequence. So this is a way to actually confirm what you learned in molecular biology. The same you can see from tetracycline. Upstream of tetracycline, which is from this direction, you have promoter P1, but surprisingly there is no ribosomal binding site features over here. But for the gene here, ROP gene, RP protein in the middle, there is a SD sequence over here. Maybe this is just not labeled as SD sequence over there, but I can't find promoter there. So sometimes the information is not complete in the gene bank. Other than that, there's transposons over here. This is how we'll make this plasmid. Transposon is a moving element, so they transfer using transposon and transfer the BLA gene using transposon, so the transposon get inserted here. So well, that's a story for another time. Next, let's confirm the RE sites. Okay, let's oops, I shouldn't have left the page. All right, let's go one more time. Let's open this in a new tab. So what we'll need is the FASTA format for the whole sequence of our PBR322 plasmid, right? Let's copy that and let's paste it here in, I use the Takara site here. There's a lot of sites for you to search precision enzymes. So we can select all right, and then you can list up the enzyme list, and then you can see there's a lot of restriction enzymes being detected, including the sequence of the restriction enzymes, which might be useful, which might be not. But for our example today, let's just look at the map again, because we have the map, just to confirm everything is in accordance with the map. we have eco R1, eco R5, BUM H1, SPH1, SAL1, right? Let's look at SPH1 and SAL1 and eco R5 maybe. So let's go back and unselect clear everything. Let's look at eco. So this is in alphabetical order, I think. So eco R5 is there, eco R1 is there. Let's look at S, was it SAL1 and SPH1? Let me move back. It's SPH1, right? SPH1. Okay. And then you can click on the enzyme list. Now you can see there's eco R5, SAL1, SPH1. There's frequency, that is the number of location for recognizing the restriction enzymes, and then the cutting position is 187, 651, 566. So SPH1 comes first before SAL1 and then eco R5. But you can see there's no eco R1 over here. However, in our map, there's an eco R1. So one explanation for this is because the size of the plasmid is 4361, and this is 4359, which means that the eco R1 site is being cut when we register the information in GenBank, so that it's not detected here. So eco R1 is where they decide the number one, apparently, right? So that's the explanation. But other than that, we have eco R5 first, and then we have SPH1, and then after that, SAL1 over there. So we can confirm that location over here. The distance between these sites, 187, for example, 651 minus 566, all right? So it's about 90 base, 90, was it 90 base pair, 90 base pair difference. So let's look at this distance over here is about 90 base pair as well, right? So you can confirm the location of the sites. All right. So normally before we start cloning, usually just look at the map. But then for today, because we've learned about database, and I want to show you something interesting that you can visualize using the GenBank, like the SD sequence of the ribosomal binding site and the promoters. So you can inspect that using the GenBank over here by just clicking at the graphics over here. You will click at FASTA over here rather than the graphics, right? So we confirm that, and next we want to find restriction enzymes that we can use. So normally we'll use tools like this. You can find, I think, restriction enzyme. If you Google, you can use other sites as well, like RE enzyme finder, for example. I just randomly put this tool. So that's from Thermo Fisher as well. You can use that because previously I think we used Thermo Fisher. There are different sites like NEB, New England database, New England Biolabs also provide their enzyme finder as well. So the most important thing when you want to clone something, first of all, you want to find the site that cuts only one, right? One frequency over here. If you have sites that cuts enzyme that cuts multiple sites, that's not useful for our cloning, because our plasmid will be split into two or more than two. So we just want to linear, right? Linear, so that it's linear rather than being circular. So we just want to cut at one side. Right? So for this purpose, when we select, let's say, the RE from all of this list, and then we submit again, you'll see that frequency 1, for example, AAT2, right? And then AAC1 is not useful because it's 2. So we can sort this by frequency so that you can see somehow it's not really sorting very well. So frequency 1, so you can choose the one that we can use AAC3, and then but we have to look at the location where we're cutting, and then ideally because we need to select the clone using ampicillin resistant gene. So we don't want any of these restriction enzymes because this will insert our DNA into ampicillin resistant genes, and that is not good for us when we want to uh, extract this from E. coli again afterwards. So ideally, if we're not using tetracycline, we can choose this one. My choice will be like EcoR1 over here because we know it's a single cut, or EcoR5 or BAMH1 over here because this is also a quite useful enzyme because I think most labs will have BAMH1 or EcoR5 or EcoR1. That's a very common research enzyme to use. So the first screen is to look at which enzyme that cuts once, and then the second thing to consider is that whether you have that enzyme or can you purchase that enzyme from the manufacturer. So like I mentioned just now, I don't know about AT1, but I know we can buy BAMH1, or for example, let's look at what else we have, like EAM1105. I don't know this. This is ECOR5 is something very common. We can use that, right? HAE also is common, but there's 11 sites, so it's not very useful. And then we have NDE1 over here, which is also quite useful, right? Next is to check. Now we check our which site is available on the plasmid site. We also have to check what is available on the opposite, the insert site, right? So because we want to use, let's say, for our assignment, we are using the spike protein. So we want to use the spike protein gene, which we can get the accession number from the previous assignment, NC45512. We copy that, and this time we can put it here. So click on FASTA again, and then let's copy the whole sequence. This is very long. All right, copy, and then let's search this. Let's just use the enzyme finder again. I copied that first. All right, let's just put that in Notepad for a while. Or we could just do this here. So for our insert, since we identify, like for example, EcoR5, EcoR1 to be our target, we can clear the selection, just choose EcoR1, EcoR5. And we have NDE as well just now. So let's find NDE1 and just search in our sequence whether it will cut that. All right. So we have 5, so that's not very useful. That's not very useful. So how do we need? How can we clone part of it? Looks like we have to find single sites as well. So single is one. So there's H I one. All right. Okay. Let's go back. Looks like I choose a bad example again this time. Nope. Okay. equal R1, equal R5. I guess it doesn't matter if we have multiple sites. So we have equal R1, equal R5, and DE1. But for the insert side, we don't have to cut it using restriction enzymes, because from the example I showed just now, you can add linkers, all right? We can add linkers to your genes. So we'll add equal R5 over here, and we add equal R5 to both ends, right? Oh, no, we can't do that as well. All right, the reason why we can't do that, because we have to cut the equal R5 so that we can join them together. So equal R5 is not a good choice. Now, which means that we go back to our map and let's identify other sites. For example, SPH1 and SPH1 and SAL1, right? Okay. All right. Finally, we have a hit, which means that we don't have SAL1 in the for our COVID gene, right? We don't have that. So we can add the linkers, right? We can add the linkers just now. Add the linkers of SAL1 based on our results here, because it's not detected here. And then we can cut it with SAL1 and cut our PBR3 to SAL1, and we can insert it in that location, right? So we can technically insert our gene of interest into our PBR322 plasmid, right? So the problem why I had a lot of difficulty here, because PBR322 plasmid is not so useful because there's not much selection of restriction enzyme sites that we can use, right? Typically for cloning, there are other examples like PUC18, right? PUC18 over here. PUC18 in this region over here, it has this region called multi-cloning site, and you have a lot of restriction enzymes here, right? ECOR1, SAC1, KPN1, SMA1, BAMH1, XBA1, SAL1, and go. So there's a lot of selection you can choose. So you can use this and check whether the glycoprotein has this or not. If it doesn't have, you can add the linkers using that sequence, and then you can cut the PUC18 and insert your gene inside, and then ligation, right? So that's how you clone it. So the cloning steps as shown here, right? This time we'll use PUC18 or 19, right? Or you can use PBR322 as well, but Simon is using 19 if I'm not mistaken. The DNA that we'll be using is the piece of DNA is the gene of a glycoprotein. You add the linkers, do a restriction enzyme on both, and then add ligation, and then to this point, right? Insert it, transform into E. coli, and then you'll get a lot of plasmids after that. So that's our objective. So for this week's your group activity will be clone the spike protein, spike glycoprotein gene into PUC19. So PUC19 is the opposite, the multi-cloning site is the opposite from PUC18. PUC19 starts with HIND3 and ends with ECOR1. PUC18 is the opposite, it's ECOR1 and then HIND3. So it's opposite. So I want you to use PUC19. Why? Just to add, it doesn't really matter because we're using a single restriction enzyme site. And then I want you to retrieve the PUC19 DNA sequences from the gene bank, identify the RE sites from the protein, the number for accession number you can get from the slide here on top, the previous activity. This is the accession number for the glycoprotein. And I want you to search for which restriction enzymes that you want to use and write the protocol of how you're going to clone the spike glycoprotein into the PUC19 for DNA sequencing. All right. So there's a hint over here for DNA sequencing. So what you need to do. So that is something that I want you to think about, right? For DNA sequencing. And then question number 4, that's a question rather than instructions. What is the benefit of sequencing after cloning rather than sequencing directly, right? You know that you can sequence a piece of DNA very easily if you know the primer, but now we are putting it into a cloning vector, all right, PUC19. So why do we do this instead of sequencing directly? All right, maybe some of you already have the experience of doing this, as you know why we are doing this instead of directly sequencing that piece of DNA. So deadline as usual, 1 week on Monday before midnight, and please discuss with your group. All right, your answers. I also realized that a lot of you are doing individual work. I would like to encourage you to do discussion because it helps students from SIT to communicate in English, and then maybe will also give you ideas about how to solve this problem. So any question before we end the class? So for information, this is also in the classroom as well. In the activity part, I think in activity, right, spike clone spike glycoprotein there. So the instructions is there. All right, okay. No question? Right, if there's no question, I think we'll end this class today. And I think there are some students who have not submitted previous assignments. I think you can submit it anyway. Please submit it, even if it's late, it's okay. If it's halfway through, it's okay. I expect a full submission. At least it will help you in your assessment for this course. Right. Thank you very much, and see you again next week for our last session for this collaborative learning. Thank you very much, and I will end the sharing of the screen and also this Zoom session. See you next week.